Welcome to Taxonomy Tuesday. My name is Chris. I'll be your host this evening. We share our finds if we find any, and we try to help each other identify them. And you could send in emails to Taxonomy Tuesday or put your name in the chat if you have something, and we'll get to you promptly. Hunter, did you want to start? You got an email in.
Sure, absolutely. Happy to.
All right.
Oh, okay. Yeah, so they came in kind of out of order or backwards, I should say. But yeah, let's just slide down to the beginning to the mushroom, if that's okay.
What do you got here? This one?
Mm-hmm. Yeah, right there. Yeah, actually, I thought there was one more picture, but this, I'm pretty sure is Pluteus cervinus. It's the deer mushroom or the shield mushroom, or sometimes called the fawn mushroom. And it grows on wood. This was growing on a decayed log, and it has very close gills, and it looks to me like they're free gills. And uh, it also looks like there's maybe some short gills, but I can't tell for sure. Maybe not. But it grows on wood in the spring, and it is edible. And I don't know how great it is. I haven't eaten it, but people do eat it. And we can move on to some more pictures of it. Here's the stipe. Nothing really, I don't know. What would you say? It seemed like there's some little flocos, perhaps, but maybe it's just the picture. I don't know. These
No, it's
pictures,
oh, go ahead.
well, they look better that way. Yeah, it's interesting. Okay. Yeah, and then these are the spores under the microscope. And so they're elliptical, would we say?
Yeah.
Yeah. And I think relatively big. Um,
Is this at 400?
yeah. Um, yes. And also maybe expanded a little bit though. Oh, I think this is only at 40 and expanded like 2 times.
Oh, like with your phone?
Mm-hmm.
Nice.
Maybe. And then let's look at the spore print. And so it's a pink spore print, and this really came in thick and dark, and it's kind of wet, but it's real pretty color.
Yeah, that's good. That turns to that like peachy brown almost, but man, that is a good thick spore print.
Yeah, it's really thick. It's on a little clear saucer, and then I covered it with a bowl, but it was, out of the woods maybe um 20 minutes before and actually it never went in a bag um or in a box or anything. It just kind of went from my hand to the car seat to the plate all this morning. So, and this was left really quickly. So this was, I put it down probably at maybe 9, and then this morning, and then this is how thick it is already now.
For any of our new folks, if you're looking for that spore print color, getting a good print will get you the better color.
Yeah, this is really deep. It's nice. And then I've got the cystidia too, which is a telling factor for Pluteus. So they say it's sort of antler shaped. And so here's one of the cystidia, which is structures on the gill edge.
Yeah, that's a beautiful photo. That's the telltale.
It's funny, it looks like it has an eye, like it's a fish or something with an eye, like he's with its mouth open, kind of. But anyways, yeah. So got that. And yeah, but there wasn't really much else out there today that I could find, but I didn't, I've been kind of resting up. But this was interesting. This was up in Wisconsin, and I've shown another picture before of puffballs growing on um a pine cone. Um, these are very old, and I apologize for the kind of, they're not that pretty, but they're growing right on, these puffballs are growing right, and this is a different kind than I've seen growing on them, a different kind of puffball. But this is basically my second pine cone that I found with puffballs growing from it. And I know puffballs are really adaptive and will grow on a lot of different substrates, but it's not something you see every day growing on pine cones. And so
Do you know what kind of pine cone that is? What tree that comes from?
I'm gonna guess a red pine of some sort.
That's
But
cool.
no, I mean, it seemed like there was a lot of a red pine. I'm not that great with trees, and I asked my husband, I was like, "Are these aren't white pine?" Because I didn't think they were. He's like, he thought they were red pine, but neither of us are experts on trees. So, uh, but yeah, I mean, it definitely wasn't. I mean, I could say a lot of things it definitely wasn't like so. But and then the next picture just kind of proves that it's a puffball. This is a typical interior of an old puffball, kind of cottony and dark colored, like you see. So yeah, that was about it. Besides a few morels in the area, but it was a little bit dry.
I wonder if that thing is like host specific.
I don't believe it is. I mean, I really think that these puffballs are opportunistic, and I mean, because the other one I have, it's a really nice picture of a puffball, and I think it's what is the one? It's like Curtisii, I think, like lycoperdon. It's one of the small sort of studded white puffballs, but it's definitely not host specific. I mean, I guess we can't prove that unless we did the microscopy and even DNA test. But it's my understanding that puffballs are not that picky. So I think it's just sort of a freak.
Cool.
Yeah, and I didn't keep it, so I couldn't do anything with it, but I thought I'd share. And then just a couple morels. Um,
And did you get the poison ivy getting them?
no, I didn't. Luckily, I thought it looked pretty though, didn't it? I'm not positive that is poison ivy, but you're probably right. It certainly could be.
I got poison ivy right now.
You
I
do.
always get, I have to, yeah, I know what it looks like. I'm pretty sure that's poison ivy.
Yeah, you're probably right. I'm not that prone to it, but um I usually do recognize it, but I also know that it can really look very different sometimes. And then there are some things that look very similar.
Some decent sized morels, huh?
Yeah, these were pretty nice. Yeah. Yeah. Yeah, they were pretty big. Yep. Yeah.
Which we got
Very.
them in my neighborhood.
Oh, good.
I don't ever see morels.
So these are most likely Morchella americana. I would suspect. That's all I have.
Nice. Thank you.
Thanks for letting me share.
Nice. Does anybody else have anything? I saw some stuff from the this past weekend's foray in the INET.
Yeah,
Check.
Chris, if you want to just go to my page.
Hopefully, Emily will bring some mycelium pictures when she's done eating too.
What was that, Brandon?
Yeah, I have some stuff on iNet. If you want to even just go to my page, I could always email them to you. But yeah, here they are right
I
here.
think some of these are yours, right?
Yep. If you want to go to that Cryptoporus, the little yellow thing on the pine tree. Yeah.
Oh,
So
cool. I love that one. So,
yeah, it's a really cool one. It's not on. I mean, it's common, I would assume, uh, but I only see it a couple times a year. Um, it loves the pitch pines in uh, the pine barrens. So I'm not totally sure on, what else it grows on, but I've only ever seen it on pitch pine, Pinus rigidus. And then, the
I've seen,
underside,
I think white pine.
okay, cool. Well, that's interesting because white pine's a hard pine, I think, and pitch pine's a soft pine.
Okay,
So
maybe
maybe,
I'm wrong. I might be
well,
wrong.
no, it just might be a conifer thing, not down to one or the other.
Yeah,
Um,
possibly. It'd be good to find out. Yeah.
But the cool thing about this is you can kind of see I've opened the lid. I should have taken a photo with the lid closed. But the common name for this one is the veiled polypore. So these obviously mature pores, um, because all that pink, those are all the spores. They have pinkish spores, and you can kind of see deposits of that pink. Um, so that exists before the veil kind of breaks, uh, which I thought was interesting.
They just
And
kind of pile up and get ready.
yeah, because Jason told me on Sunday, I found this with a couple of people, and I was like, yeah, I'm not sure if it's exact ecology or what's going on there, but I think eventually in maturity, this veil just kind of cracks open and the spores kind of fall out. But Jason told me that it's a specific beetle that will kind of bore through um, that veil. So it kind of makes sense that they're already lumped in there. So when the beetle pokes a hole in it, the spores are all ready to dump out.
Hmm.
Right. It totally relies on insects. And I had thought it was ants, but I guess I'm wrong. Yeah.
Maybe there are multiple, or maybe there are multiple, you know, things going on. Like you're finding one on white pine, and it's ants, and Maybe down here it's beetles, but yeah, it could probably be multiple things.
Yeah, that's such a cool find.
Yeah, and then there's the flip side of the veil. So pretty much the photo that we just saw, the other side of it, uh, and you can kind of see it's membranous, and it has that. It's it's. I don't even know if you would call that a veil. I do think there's vellum on whatever structure you would call this, uh, but yeah, I should have looked it up.
What if?
Like a veil. I mean, what what is vellum?
What? What?
Vellum?
Yeah, so vellum would be like veil remnants, but
Okay.
the structure that this thing has, you're familiar with it, right? It's really thick and tough. So I think there's kind of that universal veil that it does exist on this thing, but it's not necessarily the actual lid, if that makes sense.
No, I mean, this
Yeah.
is actually pretty interesting, this one, because you can see the whole tube layer and that whole empty space where in yours the spores are piling up in there.
Yeah, that is cool.
And it never gets like thin enough to break. I mean, look at how robust that thing is.
Right.
So it would make sense that it would rely on some other means. That is cool. But yeah, it looks like they hatch out of this other part almost.
Yeah, I
Yeah.
haven't seen them red like that. Only like, wait, that's cool.
Oh,
Oh, wow.
Wow, are they red under UV, the pores?
man, I had my light with me too. We could have had some fun.
I might have this thing.
Wow.
Oh my gosh. I can't believe how many there are. That's wild.
Yeah,
That's
I
an
don't think I've
incredible.
ever seen one of these, honestly.
That's
So
more
this is
how they looked when I saw them. And there was only a few. There wasn't like so many. That's so cool.
Yeah, and this is how I normally find them. And can we go back to that last photo just to kind of clarify what I mean with the universal veil versus whatever evolutionary tactic this thing developed for that. Well, I guess it's not a lid; it's more like a whatever the thing, the underside that the insects go through. Um, so like the white here, that's like universal veil. Um, and it's on the cap, it's on the underside, all of it. But yeah, the other thing, I don't know if you would consider that universal veil. It's kind of interesting.
Yeah, I, I mean, is that the orange part? Like in
That's
this
what
one?
it's what it looks like, doesn't it? I don't know. It does look like it's bursting through the orange and the red part here.
So like this, it just hasn't. I think that's what we're seeing.
I would tend to agree with that. But then does that turn white? Like what's the white stuff that I think is pretty much ubiquitous with this thing? That's definitely universal. I don't know.
I mean,
I was going to share this because I hadn't looked it up, but we were getting uh
Oh,
we were
cool.
running out of stuff to talk about.
no, it's super interesting. Yeah, that's fun. I don't.
Yeah, like what the heck is happening in this photograph?
I think it's coming out of the orange part. Or maybe that's a second one next to one that's already out.
Oh, maybe. I think that's a better idea. Like a more mature one, maybe.
And then a brand new one.
Yeah. I think that might be a good explanation for what we're looking at here, but you know, I'm not betting money on it.
I just wonder how many I've walked past like oh, that's just a glob of sap.
I don't think I don't think you would mistake it for sap. They're not. I mean, they're not huge, but you know, an inch of obscure looking. It looks kind of like a bulbous shelf mushroom or gumdrop shaped shelf mushroom. I think you would clock it as fungal.
Hmm. What else did you have there? Oh, you had this little stick I seen.
Yeah, you're gonna want to enlarge the photo. So, uh, Myco, Mycocalicium, Mycocalicium. There's another something calicium that we were considering in the field. And when I say we, Dennis Waters was there and Jason was there. So we were looking at a lot of lichen. Um, and these I've always been interested in. If anyone knows Phaeocalicium, Polyporium, that's the little fairy pins that grow on Tricaptum biforme and similar fungi. Um, but this one was teeny, tiny, um, kind of goblet shaped. And when I was talking to Dennis and Jason, they both had good ways to differentiate between two of the genera that they were considering. And this is just me being a third wheel in a conversation between them. Uh, Jason was noting that Mycocalicium, as the name suggests, is a non-lichenized fungus because a lot of these calicioid fungi are lichenized fungi. They're lichen, um, which means that it is a composite organism of fungus and an algal associate. And Jason noted that if you do if you take some pretty good photos, you'll be able to see that algal associate. So you'll see kind of a green thallus. So like the wood right up against the base of the thing will be a little bit green. And then another thing for Mycocalicium, or one of
If
the
you split
lichenized.
it open, would it have like the medulla then?
If you could split one of these things open, I would. Yeah, I would buy you several drinks. That
Ah,
they're like half a millimeter, dude. Uh, you can smash one of them, but um, I don't think either one of those would have a medulla visible, at least. But I could be wrong. Like I said, I was third wheel in a conversation between them. Um, but one of the other differentiators between the two genera that they were discussing was uh, single or two-celled spores. So are those spores septate or is there no septation in the spore? The no
mm.
septation points to Mycocalicium. The septation points to it's another genera that honestly, when I didn't really, it starts with a C. That's about all I know. And it's a bunch of syllables that uh, I've never heard together before. But yeah, you can see. So the dusty stuff, that's a different lichen up in that top left there. And those were about each one of those little clumps. I could actually measure those. Each one of those little clumps was about 2 millimeters. So all this other, black, it's all out of focus. You can't really make it out, but all these little structures, little fingers, uh, that's all I believe, non-lichenized fungus that we're talking about here, Mycocalicium.
Cool.
So tiny. Well, they don't look like they have a septa.
Yeah, and they're very small. So this is definitely at 400x, um, and they're small enough that it's not. I don't do a lot of work at 1000x. I just it's too messy. Um, but I was able to make out with the naked eye that they were not septate. Yeah, you're right, Chris.
You try using your phone Zoom a lot?
Yeah, I do.
Yeah, I know. It makes it trickier to focus though, right?
No, not really. My 2 and 3x on my phone, uh, my 3x I think takes the best photos on my phone and is able to focus really, really well.
Hmm.
And the nice thing about changing the zoom on the phone is that it doesn't mess with the dimensions of the spores, because my microscope also has a zoom feature that does mess with the dimensions of the spores.
Hmm.
But yeah, I wish.
Nice. How did you end up getting these, may I ask?
What do you mean? Like, how do I get some of it to take home?
These spores, to be more specific.
Oh, this was a smash mouth. I just,
Just plucked one off and smashed it.
yep, yep. And the funny thing about it is, like, when I say I would buy a couple drinks to section one or something, um, I couldn't find any material. All I found was spores and some like wood debris and things like that. But I couldn't find,
Oh,
yeah, because I was looking for, well, where are these spores coming from? Or, could I find differentiation in the layers of this thing? Like, does it have a medulla? Um, and I didn't find any material. All I found was spores. So, which begs the question: are these spores from that thing? Not sure.
you still have it?
Yeah.
I've done a couple of times where I've like taken like a little thing and then like glued it to another thing
Yeah,
so I can put it under there, like
that's
not
a
on a slide.
yeah, that's a good practice. That's for a lot of um, like little disco mycetes, things like Orbiliaceae. Um,
I'll bet that thing's interesting to look at intact. I wonder if it just got some sort of like coating, almost like I don't know, like a xylaria or something, like where they get in that other stage.
well, yeah, it's a good question. I wish Jason was here to answer some of them. Like I said, a third party just trying to regurgitate some other stuff I heard.
That thing's cool though. And there's no other material. It just completely smashes up into bits. And it's all spores.
Well, no, theoretically, that's not true.
No,
But
but
I just wasn't able to properly do it.
hmm, that's super cool. What else did you have in there?
Those Mycena that we found this weekend are right there. If anyone has any idea, I think we talked about inclinata as one of the only things that wasn't dried up we found. Pretty. Yeah.
I think the conversation went something like, I don't know, call it inclinata.
Did you smell them? I probably did. I had a few new members with me and I was, talking through everything with them and how to identify. I probably did. I don't remember there being a smell. Yeah, that basal mycelium was really thick and fuzzy. It was soft to the touch.
And I think by the time I saw these, a lot of the yellow in the apex of that stipe was gone. So, and the yellow is an inclinata thing, if I'm not
Yeah.
mistaken.
It's funny when I was looking at inclinata for these, I'd read that and I was thinking, oh, they really don't have that much yellow. So I guess I'm not sure how much yellow is
Yeah,
required. I mean, that's a hint
yeah,
of it. I wouldn't say they were very yellow though.
yeah. These are pretty interesting. Is that like what's going on the Is there like a lot of insect damage or something like that? Probably.
Yeah. I
Yeah.
think
Okay.
some
Yeah.
slugs got to them. That's what that looks like to me.
Yeah. Yeah, Emily found most of the mushrooms at the foray, I believe.
Well, I just followed the advice. I mean, Nina said that the when she talked to the people that own the park, they suggested going on the yellow trail. Well, we were the only people that went on the yellow trail, so we found stuff.
I'm mad. I was late and I didn't hear that, so I
Hmm.
just kind of I saw one of the Burkharts and I was like, hey, I'll tag along with y'all. Uh, but yeah, you went on the yellow trail. I would have liked to
Yeah,
see
it's
more of
such a
us go
big
that way.
park, and that's the furthest trail. Like we really did a lot of walking to get all the way out there and back
Gotcha.
in time. Yep, a very dried up version of what Hunter was showing earlier. Pluteus cervinus.
Oh yeah. Nice and dry, that one.
Mm-hmm. Frenchy.
That's like the perfect, like that's where I find them next to a log that looks like that.
Mm-hmm. It's like disintegrating.
Yeah.
I was just
Yeah.
happy we found stuff.
Yeah, it was nice. There was we had a bunch of new people at the foray,
Mm-hmm.
and we found like just enough things
Mm-hmm.
to say, look, mushrooms. These I was interested in. I don't think I've ever personally found one of these.
Really? Now I find them a lot at Parvin, and I've found them in other parks not too far from where I am in South Jersey, so maybe they're just common in this area. I mean, this isn't exactly a good representation of them. It's chewed up and dried out, but
Yeah, I was looking at these earlier. I
from the
just
top, yeah, from the top it looks just like the pheasant back.
and the stipes, is that like a pretty good representation?
Yep, with the shaggy stems. They can get really big too.
Yeah, I don't think I've ever found one of these.
And they have the serrated gill edges too.
Yeah, that was the one thing. these were missing the gills, right? For the most part.
I did not catch that.
Yeah, what was that, Brandon? It was real soft.
These were missing the gills, the
Yeah.
specimens that we found.
Yeah, there was like a tiny bit that you think was some gill, but most of it was eaten.
And you find these all the time down there, huh?
Yeah. I mean, maybe it's just on days when it's dry and there's not much else to find, but yeah, I've found them a few times. I've eaten them too. Although apparently Brandon was saying they can cause gastric upset. I haven't had a problem.
Yeah, I thought that's why they were called the train wrecker.
My
Oh, wait a
understanding
minute.
was that they were called that because they grow on the wood they used to use for the railroad tracks before they started treating them with whatever they're treating them with today.
What's the debate about? I
The
I
common name for this called the train wrecker. Why is it called that?
I agree with you. What was the other option though?
That'll wreck your insides.
Oh, okay. No, Emily's totally right on that. Yeah, I think they were banned from the like somehow banned because of that until they got the chemical that Emily mentioned. Like literally banned the mushroom.
Huh.
Yeah, because they just would decompose.
The wood, yeah.
Yeah, so well. Yeah, right. Yeah, this is so pretty. Who found this?
Well, we didn't find that one. Wow, this
Oh,
was the one we found, the chewed up old dried one. We found that this weekend at the foray.
Was that on mine? Or
Probably.
yeah,
Yeah, I see some pine needles in the background.
that's what I find it on usually.
And
Cool.
that's a great example of why common names are not to be trusted.
Right.
Yes. Um, I don't know if there's something up next, but the Wolfiporia, do you guys know that one, right? Or no?
I don't.
Yeah, I'm
Can't
not
say it. Yeah,
familiar.
can't say I do. But on Chris's note, I've found Neolentinus before, um, but not often. So I don't know. Maybe it's that area, Emily.
Could be just a Cumberland County thing.
Hmm.
There you go.
This Wolfiporia extensa, if we have time, maybe we could talk about it or look into it.
Yeah, yeah. Sorry. What were you saying about it?
Well, I'm not really from, well, what's the crust fungi that stretches like a rubber band? Do you guys have that? It's like stretchy when you peel it off the bar.
Glowy, Glowioporus,
Oh, okay. Thank you. Okay.
Dicrus, or Dicroa,
Yeah.
Dicrus, Dicrus.
Okay. Yeah, this Wolfiporia has this huge tuber, sclerotia that is eaten. And a friend of mine sent me a link to it, but I can't find a picture of the actual fruiting body. And I was thinking maybe it was the stretchy one for some reason, because I feel like I've heard the name before. It's not there, is it?
No, it is not.
Okay. It must be outside of the US, I think. Um, hmm.
Hasn't Dave talked about that before?
I feel like I've heard the name before.
Yeah, maybe we should check Myco Quebec.
Yeah, that's where I am right now.
Mm.
Thank you.
Yeah, so they have two species. Uh, yeah, extensive, right? That was the one you were talking
Yeah,
about?
yeah.
Can you pop it up or dump it in the chat?
Yeah, they're it's one of these. There's only one photo of it.
Is it the fruiting body or is it the sclerotia?
It's the fruiting body,
Good.
and it's rather indistinct.
Mm, look forward to seeing it.
Everyone, paste.
Nice.
Oh, so yeah, so it is a crust-like fungus. So
Yeah,
right?
yeah, I would call it more racupinate polypore.
It seems bizarre that it would have a terrestrial sclerotia,
Right.
which is what
Yeah.
this says.
Yeah, because when I first Googled it, I was seeing, yeah, like tuber-like things, right?
Yes.
Yeah. Uh, it's odd to think that this is in any way related to that. Um, but yeah, interesting.
Okay, thank you. Well, thank you. Thank you. Leave it up for 1 second. I just took a screenshot. I know I can go there. Thank you so much.
Yeah, no
Look at
problem.
those cystidia.
Yeah, those
Mm,
are pretty cool.
well, let me get back to the page because I would imagine that's terminal cells. Description. Oh, I didn't even. It starts
Well,
with remarks. Interesting.
I can't read that.
It's large hypogeous sclerotia, which are apparently associated by the mycelium with roots.
Oh, fascinating. Isn't that interesting?
On dead trees. Interesting.
So did you find one of these, Hunter?
No, no, a friend of mine, a mushroom friend from Wisconsin, sent me the link to Wikipedia, which really doesn't seem to, I mean, it didn't show any fruiting body growing on anything. I couldn't tell what the fruiting body looked like from that at all. I'm not sure. I don't think they included the fruiting body, but they talked about the tuber. I guess it's eaten.
Yeah,
And
I'm at the I'm at the edibility portion of this is a pretty long description, so it does seem like a pretty interesting mushroom. But edibility, inedible, sclerotium considered edible in North America, cultivated. So apparently this thing is cultivated.
oh, okay.
Huh.
And what's the natural range?
Distribution widespread across the European. No, wrong thing. Widespread across the eastern United States, southeastern Canada, the Pacific Northwest, California, and the northern Rocky Mountains. Frequency quite rare. So it's got a large distribution, but it's rare. Yeah.
Hmm, interesting. But it's
Period
also, oh, go
mid
ahead.
mid May to late autumn. So,
Okay, and not here. It didn't sound like where I am, but where you guys are, yes.
yeah, yeah, interesting.
Yeah,
But
keep your,
again, that distribution pretty much talks about all of North America, east and west of the Rocky Mountains, which is
oh, okay.
one of those delineating points. Um, so I would be unsurprised if it also exists in even like Gulf Coast states. You know,
Yeah, okay. Cool. I would like to see it. Okay, sorry. Thank you for that. Thanks for that.
Don't apologize. It's a cool thing
Yeah,
you're talking about.
it's fun. We're talking fungus here.
Yeah, but glad to know more.
So I did send
Nice.
over a couple pictures of my agar plates I've got going, but if anybody has anything else to share.
Oh, cool.
Why not? Let's check it out.
It's kind of hard to get good photos. The ones that are out right now, still growing, have condensation on the inside, so it's hard to get a good photo. But
Oh, that looks good.
so
Yeah,
that's kind of blown up, but that's about the size of a couple pieces of rice. It's supposed to be small, but I just kind of hack at it and put it in there and see what happens. So that's the blue oyster.
that is looking good, Emily. Yeah. So you're gonna wait till, or you think you're not, you put this in the refrigerator now?
I'm gonna wait and see how much more I can get on this plate. There
Oh,
should be another picture in there of another plate that's pretty much full that I have in the fridge.
nice.
Yeah, that other one.
Oh, beautiful. Nice job.
And I did cut a chunk out of it when it was still out of the fridge, when it was still growing, because something looked funny up there and I just didn't want to risk it, so I just cut that portion off.
Yeah, and so, and when you're doing the cutting, you're using what, a razor blade or?
So I'm just using a knife that I've sterilized with alcohol.
Okay, and you're not in a flow hood or a glove box or anything?
No, just moving very quickly and hasn't been a problem.
Yeah,
Um,
good. Real good.
yeah. So
Okay.
we'll see. I'll keep you guys posted as the process goes on.
Yes, too. That looks beautiful.
Thank you.
Uh,
Yeah.
if you have access, I mean, I've literally driven my stuff to the middle of a soccer field complex. But if you have access to like a big open area, that's where I do a lot of my sterile transitions and things like that.
Really?
Okay.
Yeah, yeah. Now it has noticeable. There's a noticeable difference. Um,
What about wind? You don't worry about that, or breeze or?
I think it's the things you want to keep at, well, especially like if I was going to take this and put it on, you know, I would, if this is pleurotus, I would just throw this, I'd let it get a little healthier, but throw just that portion that she took from the upper right there. Um, I know that was because of contamination
Mm-hmm.
concerns, but I would take a piece about that size and just throw it right on sawdust.
Mm-hmm.
Um, but these are the uh, transitions I'm talking about, not actually inoculating the thing. Um,
Right.
I think that's where you want to be more concerned.
Right.
But
Well, and that's also what's nice about these plates. You don't have to use the whole thing. You just take a piece of it, and if it doesn't work, you try another piece.
mm-hmm. Yeah, and when you get a healthy one, you can take a little bit of that material and pop it on
And
a
start a
new
new one right
agar.
from it. Yeah, yeah.
Exactly. Yeah, yeah.
Like the ultimate mushroom hack.
Mm-hmm. Yeah.
So yeah, Hunter, when you get your morels going, that'll be exciting.
Yeah, well,
Yeah,
that's
hopefully.
a different animal we're talking about there.
True.
Huh?
Yeah, but at least the mycelium seems pretty possible.
Right.
Yeah,
And I came a little bit late to this meeting. I understood that we were talking about cultivation of mushrooms and you're doing your morel thing. Um, but what got us there? Was there like, do you have successful mycelium growing on a plate or something? Or
I have dabbled in that before, but no, I what I said was that I I I did it all very poorly. So I, instead of just putting a few spores, I let the whole mushroom drop like all its spores in the agar, and and then I I wrapped it, but then I forgot all about it. And when I checked on it, well, when I like remembered and saw it, it was like I think completely colonized with trichoderma. They're I don't think it was worth trying to save, but I am going to try again if I can find some more morels. I think I keep putting them in the refrigerator, which I think is not a good idea if I want to use them for spores. So I have to remember to keep one, keep one out and get it back home to do this with,
So,
because they're no longer in my zone. But then we had a cultivation curiosity episode on doing this with morels, and apparently there's been success in growing the mycelium, but that's it sounds like as far as it's gotten with the natural American morels or Eastern morels.
and is there, because when I find a mushroom that I want to create a culture of, uh, I use the flesh of the mushroom.
Yeah, we were
Uh,
talking about that a bit too, because Emily started those cultures, right? Were from inner tissue
Yep,
of the
just
mushroom.
in the inner tissue, right?
Yeah, right. And so well, I mean, morels are hollow, so there is no inner tissue. Maybe, but this is how it was done in this. And I have experimented with that as well, but this is not with morels because, like I just said, there is no inner tissue. But this is how they showed it in our class, our episode.
well,
Um,
if we're doing experiments, and now I want to get some plates going, uh, I would try. Yeah, it sounded like you did a interesting. I came right on when you were talking about a grate, or
yeah, in the video, we watched this video. There's one on YouTube about it, but in the video, the guy who did it had they were like maybe like cooling racks, I would call
yeah,
them.
yeah. That's what
Yeah,
I was in my head. That's what I was kind of thinking.
good. Yeah, and so he had those propped up over tin foil in a like on another baking sheet or something like that, and then he covered the whole thing with a tote, but he would lay the morels on the grate so that they're so unlike my thick heavy Pluteus print, it wouldn't be sweaty, and you know what I mean, and he thought that would
Yeah,
reduce
yeah.
contamination as
But
well.
but I would think, even if it's not the innermost flesh, because you want the innermost flesh because that's going to be the most sterile flesh, right? That's the
Right.
whole reason you go for the innermost.
Okay.
But if we're, if you're trying to grow something on agar, I think you want some material on there. So even if
Okay.
it's even if it's not, because yeah, obviously these things are hollow, but
Right.
if you can get, you know, even those ribs, like maybe remove the ribs, and if you can get some kind of like, because you don't have,
Well,
I don't know.
I mean, when you do that, you're doing what's called cloning, correct? Yeah.
Yes.
Yeah,
And so, but I think when you do it from spores, you're not, I guess. But
well, it's the same genetic material, right? Well, I guess one's,
Well, from spores, you now have, whatever diversity there, and then
yeah,
you have potentially a plate full of many, many mating pairs,
right.
and you're kind of letting you know, the rest of those processes ensue which is why, like, a lot of times when people are going from spores, they're trying to like lessen the numbers so they can purposefully breed uh, uh, monokaryotic uh, plates. So they'll grow like they'll they'll try to weasel it down to as little of spores as possible, and as soon as they germinate, they're pulling them off and transferring them onto their own clean plates, unmated, and then they can just like kind of then mate them one after another. And they, if you got something that you can fruit, you can potentially get many different variations.
which, for this these purposes, that's not necessarily what you're going for. I wouldn't assume, Hunter. We're just trying to grow these things.
Yeah, I mean, you
Well,
probably would want a clone.
I mean, with a morel, it's we're not going to be able to just throw it on any. I mean, the next step is not really known because it's a mycorrhizal mushroom. So, so perhaps doing it this way, maybe. Well, I mean, I can only assume that it hasn't worked the other way, or I would think that guy who led the cultivation curiosities would have. I don't remember him saying like you could also try this with tissue. Um,
I've
but
heard of people doing slurries with morels,
right.
and they just basically take a whole fruiting body and throw it in a blender with some uh, like a little bit of like molasses or something, like slightly sugary, and then they dump it into a big thing of water, and then they just go dump that everywhere, and they just hope for the best.
Yes, very true. Right. Um, that is so true, and just the yes, that is true. But how successful, I don't know. I mean, I'm not arguing that. I'm just saying that I was just trying what we were encouraged to do in the class because then he encouraged us to all try it. So that's all. But I maybe I'll try some inner stipe of tissue.
So I have a question, Hunter, because I know that we discussed a lot of different things in Cultivation Curiosity last year. I know we also touched on liquid cultures. Um,
Right.
and so before I started the process that I'm doing now, I'd kind of looked into both methods, and my understanding was that liquid culture works better with spores, but the plates work better with the tissue. Um, and I don't really remember why what I had read that I came to that conclusion. Did we talk about the agar plates? I might have missed that one from Cultivation Curiosity last year because I know we talked about the liquid culture.
I actually think it was I want to say it was the very first or second one. Um, but it was with I can't remember his name. He led it. He
Probably Jake.
Yeah, Jake.
Yeah.
And then yeah, and I mean I think it was like his first one at least. Um, but but he shared this video that someone else did on it, and yeah, I know we talked about. I actually only attended maybe three of them or four or
Mhm.
three, so I don't know for sure what was talked about, but yeah, we talked about liquid culture at some point, and yeah, so I don't know. I haven't been successful with that at all. Has anyone else?
No, I haven't tried that yet.
Yeah, I haven't accomplished anything yet.
Does anyone have a link to the video that they shared?
I'll look.
Thanks.
Yeah, I think all of those you'd have to ask uh, for them because they weren't put up on YouTube
Yeah, we weren't able to put them up on
because
YouTube
they were
because,
videos in
yeah,
them.
right, because we shared other people's videos.
Right, and I'm looking for the other person's video that they shared.
Right. I might have a copy somewhere in my email. I'll take a look too.
I do
Yeah,
have the one that um can I share it here or no?
you could
That's
pop it in. You could
okay.
pop it in the chat for
It's
sure.
not the original video, it's the one where Jake shows it. And there's sort of a sound issue at first, um but yeah, it's in there. Hopefully that'll work.
Nice. I'll check that out
Yeah,
after the meeting. Thank you.
yes, my pleasure.
And then we've been on this photo for a while. Whose is this?
Th- This is some mycelium I found in my garden.
Wow,
That's
It was
lovely.
it was plentiful.
that's great.
I pulled back the pile of leaves, and you could see there's even more down the right hand side that I kept pulling back after this.
Oh my gosh, I did the exact same thing just today and I saw it, but I was like rushing and didn't really pay attention. Do you ever put like spent blacks in there or anything, or have you grown anything in there?
Nah, this is just whatever
Natural.
showed up.
Yeah.
But
Cool.
this is you could see how big this one good patch is here. I mean, this thing's got to be, I don't know, probably like almost 4 feet long, and then running down the rows like right in front of the rake there. That's like I had the wood chips all in between my rows here. So it's like running all down the rows as well. I'm just waiting for stuff to pop up, really.
Yeah, I wonder what it is.
Could be anything. I don't know how to, just tell from the mycelium. That's for
Right.
sure.
And there's no guarantee it'll fruit either.
No, no, no. It's just
Or
I'm
or
glad it's
when the
eating my wood chips, quite frankly.
or when the last time it fruited was.
Well, there's never really been a good fruiting on these wood chips. These are only like they spent all last year there, but I got them it was still like winter. So I haven't had, like, if they're going to do anything, I would think that this would be the first year that it would.
Yeah, it could be arbuscular fungi, right?
Oh, I don't know.
With no fruiting body.
But it's I assume it's in these, like that pile, like where this long stick is in the back, like that's like a 1 foot tall mound of wood chips. And that goes all the way around uh this whole garden plot of mine. So that's 50 linear feet of like at least like a foot deep of wood chips.
Wow.
So I'm something's probably gonna happen, and it's probably gonna be spectacular eventually.
Maybe inky caps.
Who knows? Who knows? But you know, being that it's in the yard, I'll probably get some good photos when it does. And then I found uh, these the other day in Shark River. Or no, these were at the reservoir, the Manasquan Reservoir. And they seem to have like a bit of a ring there. And I still have one. I collected one, but I just haven't done anything with it yet.
Weren't we looking at something like this last week? Or
What do we think? Maybe Carinomyces?
A Grossaby?
Yeah, could be a Grossaby.
Yeah, just a tiny little clump of them in the, right in this broken apart, disintegrating log here. But it's really like all I found, honestly, lately. Not too much going on out there yet.
Well, we're supposed to get 4 days of rain this week, so
I know I keep looking at it, but it's like, oh, it's gonna rain like for like an hour here, and then maybe an hour tomorrow. I want it to rain all day.
yeah.
Does anybody else have anything out there? Are we gonna wait for the rains to come and give us more things to see and talk about?
I'll share a couple things. I'll put them in the
Yay.
chat. They're from a while ago, and they're not that interesting, but we're talking about Pluteus. We're talking about, well, anything that we can find, huh?
It is dry out there.
One.
Yes, I am human. What is
Other
that?
one. All right, so this is Reticularia, lycoperdon. It's super common. It's a slime mold. It's not a fungus. But as confident as I am with Reticularia lycoperdon, this one threw me. I've never seen them present this yellow. Maybe I caught it at a weird stage of development, but well, that's definitely what happened. Yeah, that's what it is. I double checked with a couple people, and they agree. But yeah, the size and the color really threw me off. But it makes sense. It's an immature. It's kind of in that in between stage of plasmodium and forming a true mature fruiting body. Um,
It's cool.
Are they usually bigger? Are you saying?
they get a little bit bigger. You can see with the penny there, it's a little small for Reticularia. They're normally a couple inches, and this is maybe 3 centimeters, so not even two. Um, I guess it's not that big of a distinction, but again, theoretically, this thing is going to get a little bit bigger, lose its coloration, and you can see that kind of mat of yellow. These things like to leave, what do they call it? Slug trails. So you can often see Reticularia leaving some evidence that it moved from one stage to its final placement as a mature fruiting body by quote unquote slug trails.
Weird. And that like it'll be like a dried mass next to it type of deal, or it'll be like
Yeah,
this,
some
like egg white next to the egg yolk.
a little bit in between that. It's like the ghost of an egg white, if that makes sense.
We
Or yeah,
Yeah, I don't know if I've ever seen one of those. Or maybe
It's
I called it some other thing.
it's definitely a spring thing. Um, I've seen it in the spring in bottomlands. And
and
what
that's a good point, Hunter, because I didn't call this like I said. I was confused about what this was based on the spore color here. I thought this was a Fuliga, uh, and a species not Fuliga septica,
oh yeah.
but Fuligo ludio lutescens, or something alluding to the yellow Fuligo that isn't septica. Um,
Right.
but but I was told that those spores appear that color just because they're mature and they'll change that color a little bit. Uh, so
Cool.
That's awesome.
Yeah, that's a cool find. And the texture is really weird on those.
yeah, yeah. You want to talk about? We were talking about eggs. The outer shell. They do. They have a shell. It's like an eggshell kind of. It's less uh, more brittle than a normal eggshell, but in maturity, you kind of have to crack these things open.
But you can often
Yeah,
like push into them a little bit.
it seems thinner than the uh, Fuligo. Usually their shell's a bit thicker. Like you could almost see the mass past the shell,
Yes.
which is
Yep.
cool.
And if you look at Jan's comment down here, she's kind of summarizing all the things we've been talking about. So, uh, and she mentions Hunter, like you said, seasonality, which is something that don't often consider when I'm looking at slime molds, but I definitely should more often because she agrees seasonality makes way more sense here for Reticularia. But you can also find the last year's fruits of that Fuligo that I was talking about this year, but they would look much different than this.
Hmm, nice. Maybe you'll get one of those as a pet.
I don't think they show up too often in moist chamber cultures. These massive fruiting bodies, the things you find in moist chamber cultures are the things that even in maturity end up being smallish. Um, but yeah, I was talking about that thing small. The other reticularia is even bigger. You're not going to get a Fuligo, I don't think, to fruit and maturity in a moist chamber culture or a
You
or
probably need a bit more log or whatever.
just more
Then
space for it to actually mature. I mean, if I try to fruit that in that reticularia, like even if the plasmodium made it to the maturity stage, it's breaking out of the petri dish or whatever item I'm using to mature it. It's bigger than that. Yeah, and then this is just an interesting Pluteus that I have. I dried it. Maybe I'll work on it, but it's probably something in the cervinus group, for lack of a better word. Um, Even though it does have these really interesting, uh, I don't know, tufts of hair.
Saber-like. That's
Yeah,
cool.
they're like elongated scabers, or I don't know.
That's interesting. And mine was um it seemed like it might have had hairs, but they weren't dark. But I think, oh, look at that.
Yeah,
Nice.
and you might look at that and say, well, that's probably not Pluteus cervinus, but there's plenty of observations on even Quebec and uh, respective places that, yeah, they do have just tufts of hair on them.
And its type is really a lot.
Yeah, and over, I I don't even take photos of Pluteus anymore, because I look at it and I go, okay, buddy, that's Pluteus. If it's a maybe Petacetus type thing, yeah, maybe I'll do the work on it and put a Petacetus type name on it. But these Cervinus, man, they're so morphologically diverse, and the micro doesn't help all that much. You have to do like perfect micro on it to differentiate between what 1 to 2 microns difference between the spores, and um, you have to really get a great Roman aqueduct cross section of the thing to determine how large and where those cystidia are placed. So I've done it enough times to know that I ultimately always just kind of call these things Pluteus. Uh, so, but if anyone has a suggestion that isn't, this might not be a common Pluteus, I'd love to hear it.
Yeah, I mean, like you said, that I mean, all the hairiness could just be, morphological from where it was sitting, in a windier area or whatever. Maybe it would have a shinier cap or something. But it's cool.
I remember, was it a year or two ago when we had a lecture by Alfredo Justo through the NYMS program over the winter, that he showed two pictures featuring multiple specimens of Pluteus, and he asked how many species we thought there were in the picture that showed, like, great morphological, variants, and people say, oh, they must be all different. That was all the same species. And at the same time, the second picture showed things that were the same, and they were whole different species.
Right. Yeah. So,
And that's section Pluteus for you. Okay. Good luck. I know. Forget about microscopy. Just better, save this and submit for sequencing and get your name 5 months later, something like that. And you'll know for sure, as opposed to, like, second guessing.
Are they something, Igor, that I know that you've even said that some stuff doesn't, translate well with the DNA. Do the Pluteus take well to that?
I don't know. I mean, I know nothing about this genus. I don't see Pluteus much. And that's a problem. When you don't see mushrooms anymore, it doesn't make sense to learn anything about them because you cannot practice your knowledge. And okay, you see this mushroom once a year, and you won't see it again until 5 years later. Any knowledge that you might have about the species at the end of those 5 years is probably gone. I mean, it just doesn't stay in my head. Um, Even when my memory was much better, I still struggled, with things like that. If you don't repeatedly visit the subject and review the details, you just don't remember these things. They just disappear naturally.
For
I,
sure.
I don't know. I was looking on the Mo earlier today, and I saw these very attractive Russula that was like dark green in color, and I'm like, geez, somebody proposed the name because apparently it had a DNA sequence. It was atro something, I forget. I'll probably find it.
You're
And I blasted the sequence, and yes, there were multiple examples of accessions that were 99.5 or 6% similar from all over the world. Europe, China, and it was all called the same name. Like, geez, do we really know if this is the same thing or it just happens to be a different species? And ITS just fails to resolve this particular cluster of Russulas. Yeah.
Well, then anytime that there's any ambiguity when I get something sequenced, it always comes back to, was the type specimen sequenced, or do we have a clear understanding of, why that name is that name?
Yeah. But even then, there's always a cautionary tale that your ITS or maybe any other locus that's just slightly superior to ITS for barcoding purposes is not going to deliver you a perfect answer all the time. Maybe 75% of the time, but not all the time. So we've seen enough in Agaricus that different species that are clearly different morphologically have an ITS sequence that is only like one or two base pairs away from each other. Otherwise, you just call them the same by ITS, but they are different. So, how many genera are plagued by this problem? Like Cypherella. I just had discussion about something similar on the Mo maybe 2 weeks ago about the Cypherella, where types were sequenced and some names were synonymized potentially, being the same species, but nobody knows to certainty now. I don't know what to tell you. I mean, you just grab the data that you have in hand, and that's your best proposal until further notice. And you
Well,
just call it gospel at the time being and just move on.
yeah. To